Hello, everyone. I'm Yi-Fei Huang at Pennsylvania State University. Today, I'm very excited to discuss how to infer genomic determinants of positive selection using a novel statistical method developed in my lab. So our new method is based on the classic MK test. The MK test aims to detect genes under positive selection. The basic idea of the MK test is to compare the levels of sequence divergence between species and the levels of polymorphisms within a species between non-synonymous sites and synonymous sites in the same gene. So in the contingency table, we have the level of divergence here and the level of polymorphism here. We have four numbers, dn, ds, pn, ps for non-synonymous sites and synonymous sites. And Kimmel's neutral theory, the dn-pn ratio should be equal to ds-ps ratio if there is no positive selection. On the other hand, if a gene contributes to adaptation, the dn-pn ratio might be greater than the ds-ps ratio because a significant proportion of the dn might be driven by positive selection. After the publication of the original MK test paper in 1991, people have developed many extensions of this powerful framework, which have been listed here. Using the MK test and its extension, people have identified numerous genomic features associated with high adaptation rate. Those features include immune response genes, sex-related genes, among many others. You can find a really nice review paper about the MK test here. Very often, we apply the MK test to a group of genes with similar functions or a group of sites in different genes with similar functions. And I call those things genomic features. We very often apply the MK test to genomic features instead of individual genes because very often we have small number of polymorphisms in a single gene. So we have to pull genes together to improve the statistical power. Using this framework, we can study which genomic features might contribute to adaptive evolution. However, if multiple genomic features are correlated with each other, it's actually difficult to distinguish between direct effects and indirect effects of the genomic features on adaptation rate. I will use a concrete example to discuss why this is the case. Let's say if you use MK test, you might see that high tissue specificity of a gene is associated with high adaptation rate. This association can possibly be explained by the weaker pleiotropic effect of genes with high tissue specificity. However, we know that high tissue specificity is associated or correlated with expression level. Tissue-specific genes with high tissue specificity might be also lowly expressed. Then if we use expression data and investigate the rate of adaptation in lowly expressed and highly expressed genes, we might again see an association of low expression level with high adaptation rate. If we see such a pattern, then there are at least three possible causal explanations of the same observation. The first causal explanation is that possibly high tissue specificity causes high adaptation rate, whereas low expression level has no direct effect on high adaptation rate. If this is true, we can still see an association of low expression level with high adaptation rate here because of the correlation between high tissue specificity and low expression level. So the observation here is indirect. It does not imply causal relationship. That's the first plausible explanation. The second plausible explanation is that low expression level might cause high adaptation rate. So that's the opposite case of explanation one. The third possible explanation is that both tissue specificity and expression level might have direct effects on adaptation rate. So you can see that the three causal explanations have very different biological meanings, but they imply the same pattern of association here. So it's difficult to distinguish between the three plausible explanations. In this talk, I want to infer direct effects of genomic features on adaptation rate. So basically, I want to distinguish between these three plausible explanations. It's not trivial, and we need a novel statistical framework because previous methods simply don't work. The new method is based on GRM, or generalized linear model. Generalized linear model is the fundamental machinery for covariate adjustment. It has been widely used to infer direct effects of features on some response variables. And this is the general idea of GRM. On the left-hand side, we have multiple features. Those features are possibly genomic features of interest. On the other hand, we have the response variable. The response variable could be anything we are interested in, such as something like substitution rate. And using the GRM, the goal is to infer a regression coefficient beta for each feature. If we can include all the relevant features in the same analysis, then beta actually indicates the direct effect of feature. The reason is that if we include all the relevant features here, the estimation of beta actually control for the effects of other features. So if we have all the relevant features, a reasonable inverse link function here, and a realistic likelihood function here, we can infer the direct effects of features on a response variable. So that's the basic idea of GRM. Then the idea is whether we can build something similar for the MK test. If we can do that, we will be able to estimate a regression coefficient for each genomic feature of interest, and the regression coefficient might indicate the direct effect of the feature on adaptation rate. And it turns out that it's possible. We just need a novel statistical framework. We developed such a statistical framework. We call it the MK regression. It combines a GRM and an MK-based likelihood function. So it has two components, the GRM and MK-based likelihood function. In the GRM, the goal is to infer regression coefficient beta for each genomic feature of interest, and the beta indicates the direct effect of each genomic feature on the rate of adaptive evolution at a protein site. And I sent the omega A, the rate of adaptive evolution here, with some other parameters to an MK-based likelihood function. This allows us to infer regression coefficients here by maximizing a likelihood function. So that's a basic idea of the MK regression. It's a combination of GRM and MK with likelihood function. So let's dive into the technical details. The first part of the MK regression is a generalized linear model for adaptive evolution. And let's denote omega A as the relative rate of adaptive substitutions at functional site J. So it's a functional site in protein coding genes. In the MK regression, we assume omega A in a site-wise manner is a linear combination of local genomic features followed by an exponential transformation, which basically means that we assume the rate of adaptation at a functional site J is a linear combination of genomic features followed by an exponential transformation here. And the key is that we have regression coefficient beta for each genomic feature, okay? So this part of the model links genomic features to the rate of adaptation at a site. In the second part, we have a similar GRM for polymorphic rate at functional sites. We are not really interested in the functional relationship between genomic features and the polymorphic rate, but we have to model the relationship so that we can control for the effect of genomic features on negative selection. Okay, then we can send omega A and P func from the GRM to a likelihood function. In the likelihood function, we use JC69 substitution model to describe sequence divergence between species. Also, we use the binomial distribution to describe the generation of polymorphism data. And we design those distributions in such a way that they can be interpreted as a basically MK test. So the MK regression estimates the regression coefficient beta I for each genomic feature I. So that's the idea of the MK regression. We first conducted a simulation experiment to validate the MK regression. To do so, we include two genomic features, and we assume that the two genomic features in the simulation experiment could be correlated. And we have correlation coefficient on the x-axis here. Then we generate divergence and polymorphism data based on an evolutionary model motivated by the MK test. After that, we apply two versions of the MK regression to the same simulation data. We first apply the so-called simple MK regression, where we analyze features separately. In the simple MK regression, we do not try to control for the effects of other features. We actually try to see what could be wrong if we only analyze one feature at a time. We also apply the MK regression to all features jointly. We call it the multiple MK regression. In the multiple MK regression, we can potentially control for confounding features. And the true regression coefficients for the two features are 1 and 0 respectively. And we can see that in the simple MK regression, we could get highly biased estimates of regression coefficients if the correlation between features is very strong. On the other hand, in the multiple MK regression, because we analyze all the features simultaneously, and because MK regression can control for confounding features, we can see that the estimates of the coefficients agree with the true coefficients very well. So this is promising, suggesting that indeed MK regression can potentially infer the direct effects of genomic features on adaptation rate. Then we applied the multiple MK regression and the simple MK regression to empirical data. We focus on genomic determinants of positive selection in chimpanzee protein coding genes. Again, we applied the simple MK regression to protein coding genes in the chimpanzee genome. We analyzed the 6 genomic features. So genomic features include documentation rate, recombination rate, residue exposure level, expression level, tissue specificity, and protein-protein interaction degree. And those are the estimated regression coefficients in the simple MK regression. Remember that right now we are trying to infer one genomic feature at a time. We also applied the multiple MK regression, where we analyzed all the 6 genomic features jointly. And when we compare the estimated regression coefficients between the simple and the multiple MK regression models, we can see clear differences. For some of the features, yes, the regression coefficients are very close, but for some other features, they are very different. For instance, in the multiple MK regression, we see that PPI degree has no direct effect on adaptation rate in the multiple MK regression, but that's not the case in the simple MK regression. The explanation is that PPI degree probably has no effect on adaptation rate. However, it's correlated with some other genomic features. That's why in the simple MK regression, you see a significant positive association. And this is not, it's not due to the direct effect of PPI on adaptation rate. We also see that in the multiple MK regression, expression level and tissue specificity have positive effects on adaptation rate. That's not the case in the simple MK regression, possibly because of the strong correlation between gene expression level and tissue specificity. Indeed, when we look at the correlation between the 6 genomic features, we can clearly see strong correlations between expression level, tissue specificity, and PPI degree. That's why they have different regression coefficients between the simple MK regression and the multiple MK regression, because the multiple MK regression can control for the effects of other features when it estimates regression coefficient. We believe the multiple MK regression can give us better results. So the conclusion here is that PPI degree probably has no effect on adaptation rate, but gene expression level and tissue specificity both have positive effects on adaptation rate. We also explored other genomic features that might contribute to a higher adaptation rate. And one thing we observe is that metabolic genes might have a higher adaptation rate than non-metabolic genes after controlling for other genomic features. This is based on the multiple MK regression. We can clearly see a strong effect of metabolic genes on adaptation rate indicated here. And this is true after controlling for the effects of other genomic features. So in summary, the MK regression is a powerful framework for inferring direct effects of genomic features on adaptation rate. It elucidated new genomic determinants of adaptive evolution in chimpanzee genes. Those new genomic determinants include gene expression levels and metabolic genes. Thank you. I'm happy to take any question.